Hello children, we are discussing the chapter 2, Microorganisms, Friends and Foe. We have discussed various topics in our previous videos of this chapter, like what are microorganisms. The organisms which we cannot be able to see by our naked eyes are known as microorganisms. The first microorganism was discovered by Anton von Leeuwenhoek in 1674 by his first compound microscope. Also, what is microbiology? The study of microorganisms is known as microbiology. Who is a microbiologist? The scientist who studies the microbiology, who studies the microorganisms, is known as a microbiologist. After that, we discussed certain classes, major classes of the microorganisms like bacteria, fungi, algae, protozoa and viruses, in which we have discussed certain things about the viruses, that viruses are considered as living as well as non-living. Viruses do not have their metabolism, whereas living organisms have their own metabolism. Viruses only possess a genetic material, either DNA or RNA, over a protein coat known as capsid, whereas living organism has a structure of cell. Living organisms are made up of cells. So that's a basic difference between the viruses and the living organisms. After that, we discussed the useful microorganisms. Microorganisms are used in the preparation of various food items, like they are used in the liquor and the wine industry, and they perform a process named as fermentation. What is fermentation? The production of alcohol from the carbohydrates due to anaerobic respiration is known as fermentation. Microorganisms are also used in the formation of curd. They are also helpful in the formation of curd. Lactobacillus bacteria converts the lactose sugar into lactic acid, and in the presence of that lactic acid, the casein protein present in the milk coagulates and form curd. Next, microorganisms are used in the preparation of tea, coffee, tobacco, etc. Microorganisms are used in the rotting of jute fibers. They are used in the tanning of leathers, that is conversion of animal skin into leather. Rather than that, the microorganisms are used in the medicinal use, like microorganisms are used to produce antibiotics. What are antibiotics? Antibiotics are the chemicals which we obtain from the microorganisms, and those chemicals are used against the microorganisms. Which microorganisms? The disease-causing microorganisms known as pathogens. The first antibiotic discovered was penicillin from a fungi, Penicillium notatum. The first antibiotic, penicillin, was discovered by Alexander Fleming. Then second topic we have discussed in the medicinal use, that is the production of vaccines. The microorganisms in the dead or weakened form are used as a vaccine, as an injection, which boost up the immunity of a human being. So that's the usefulness in the medicinal use of various microorganisms. Rather than these, microorganisms are used in the bakery industry and certain other in the formation of food, like certain seaweeds and certain algae like chlorella are eaten up directly as a food item. Next lecture we had studied the agar-agar material is obtained from the various algae like Giladium and Gracilaria, and that agar-agar, that is the jelly-like substance, is used in the preparation of jellies, ice creams, squashes and various puddings. The topic which we are discussing now, that the earlier topics are the half of this chapter. The later half is composed of the harmful organisms, harmful effect of microorganisms. We are discussing that, and in that we are studying diseases. In my previous video, I'll explain you the diseases caused by microorganisms in humans. Various microorganisms known as pathogens which cause disease, cause certain kind of diseases which may harm a human being. These diseases are differentiated on the basis of their causative agent and its group. Some diseases are viral diseases, which are caused by the viruses like common cold, corona, AIDS. Second is bacterial diseases. The disease is caused by the bacteria like TB, tuberculosis. It is caused by a bacterium known as Mycobacterium tuberculosis. Next, second bacterial disease, the example of bacterial disease is cholera. It is caused by a bacteria which is comma-shaped Vibrio cholerae. Vibrio means comma-shaped bacteria. Next, third, fungal disease. In human being, fungus cause usually the ringworm. That is known as athlete's foot. So that's a disease which is caused by a fungus. Protozoa cause certain disease like malaria. Malaria is caused by a protozoa whose name is Plasmodium. Kala Azar and sleeping sickness, Trypanosoma gambiense. Sleeping sickness, okay, and sickness. So these are the diseases. These are the example of the diseases which are caused to humans by microorganisms. Next, in the same topic, we have discussed the various means by which the communicable diseases are spread. Now, first we'll discuss what are communicable diseases. The term communicable. Communication means to communicate something, to share the views. That's the general communication. So, communicable, a term taken from there. Communicable disease means the diseases which can spread or which may spread from a patient, from an afflicted person to a healthy person by direct contact or by physical contact is known as or are known as communicable diseases. Like malaria, corona, these are communicable diseases. And those communicable diseases are spread from a patient to a healthy person by various means. The first one is airborne diseases. The diseases which can spread from a patient to a healthy person through air, like TB, common cold. These are the two examples of the diseases which spread through air. Corona, it also spread through air. Second, waterborne diseases. The diseases which spreads from a patient to a healthy person by means of infected or polluted water. Example, typhoid, dysentery, cholera. These are the examples of certain waterborne diseases. Third is food, through polluted food. That also cause various kind of diseases. The example is typhoid. That is usually occurred to a person. The bacteria of the typhoid enters into the body of a person due to infected or polluted food. Next, fourth, through vectors. First, we'll discuss what are vectors. Vectors are the living organisms which spreads a disease from a patient to a healthy person, which carries the disease, which acts as a carrier of the disease. They carry the disease from a patient to a healthy person. So those are diseases, those kind of diseases are known as vector-borne diseases. And the vectors are the living organisms which spreads a causative agent, which spreads a pathogen from a patient to a healthy person. Those are known as vectors. Usually the vectors are mosquitoes, which which communicate the diseases like dengue, malaria, chikungunya. These diseases are spread through vectors, which is mosquito. Certain houseflies also spreads various diseases like typhoid, cholera. That disease is also spread by houseflies. Next, rats. Rats usually spreads plague. Next, dogs. Rabid dogs usually spreads rabies. So these are certain vector-borne diseases along with their vectors. Next, in the very same topic, we have discussed how a housefly communicates or spreads a disease. Usually, houseflies goes to an open area, sits on an infected material, take the infected spores, and then goes to a fresh food item, fresh drinkable item or fresh food item. So the housefly takes those spores, takes those pathogens from the infected place and takes to the healthy place. How does mosquito spread the disease or communicate the disease? So let us suppose that I have the patient of malaria. If a mosquito stings my skin and sucks my blood for the protein requirement, usually why mosquitoes require blood? To accomplish their protein need, they need human blood. So when for the protein they suck the human blood, they take the plasmodium as well. Blood contains the plasmodium. If I am suffering from malaria, then my blood contains plasmodium. And when the same insect sucks the blood of a healthy person, the plasmodium spores, the plasmodium cells are transferred. And that's how the malaria is transferred through mosquitoes. So these are the topics which we had discussed earlier. Now we discuss the preventive measures against the various diseases, various communicable diseases especially. How we can prevent ourselves from the communicable diseases. First of all, we must get the essential vaccines. Vaccines boost up our immunity, and by that immunity, we can resist a disease. We can fight against a particular disease. So, for the immunity booster, we must be vaccinated properly. Next, we must have to eat fresh and hygienic food. We must have to wash the vegetables and fruits what we are bringing from the market. Next, we have to drink clean and hygienic water. Personal hygiene must be maintained. Proper sanitation of the community is to be done so that no vectors can be produced, can be breed down so that they can spread a particular disease. Do not defecate in open. Do not go for the urination or the defecation in the open area. Next, if you are sleeping in the open area, then use the wire nets or mosquito nets so that mosquitoes cannot be able to bite you. Use the mosquito repellent creams like Odomos so that mosquitoes not bite you and you remain prevented from chikungunya, dengue and malaria, etc. Next, do not eat stale food. Stale food means open food, which is present on the various stalls in the open area. Keep the patient which is already suffered from a communicable disease like TB in isolation. You must have seen, if you ever visit a hospital, that the isolation wards are there for the TB patients, as TB is communicable disease through air. If you go near a TB patient, then you will also, or there are chances that you may suffer from the TB very easily. So that isolation wards are set up in the various hospitals for the TB patients. So these are certain, preventive measures which can prevent you from various diseases. The various pathogens also cause diseases in various animals. Like cattle and the poultry farming are very much affected by these pathogens, bacteria and the viruses. Certain bacterial diseases in the cattle and the poultry farming is anthrax. Anthrax is caused by a bacteria named as Bacillus anthracis, and that bacteria was discovered by Robert Koch. R-O-B-E-R-T, Robert Koch, K-O-C-H. Robert Koch was the scientist who discovered the bacteria Bacillus anthracis, which caused the disease anthrax. TB, cholera. There is also a poultry cholera or fowl cholera, which is usually occurs in the poultry farming. Next, certain viral disease like foot and mouth disease, fowl pox, bird flu. These are certain viral diseases which occurs to the animals like cattle. Foot and mouth occurs to usually cattle, and fowl pox and bird flu usually occur in the poultry farming, in the growth of chicks and peas, chicks and the fowls, etc., and the ducks, etc. Next, certain microorganisms also cause various kind of diseases in the plants. There are various diseases on the basis of the class of microorganisms, like the disease caused by bacteria known as bacterial diseases. The disease caused by viruses or fungus are known as viral and fungal diseases respectively. For example, the blight disease. Blight disease usually occurs in the wheat or in the rice, as well as may occur in the potato. That caused because of a bacteria. Next, certain diseases like rust, rot, smut, ticka, wilt, those are the disease examples of fungus disease, fungal disease. These diseases are caused by a fungus. These diseases occurred in various kind of plants like sugarcane, groundnut, chickpea, etc. Next, the example of viral disease in case of plants is tobacco mosaic virus or yellow vein mosaic virus. Tobacco mosaic virus usually occurs on the name of the plant tobacco, and yellow vein mosaic virus usually occur in the mustard crop or in a wheat crop as well. So these are the diseases which caused in the animals as well as in plants. Second harmful effect of microorganisms is food poisoning. Certain microorganisms that grow on the food makes the food poisonous, and that is known as food poisoning. You know what happens with the microorganisms, especially the fungi and bacteria, grows on the food very quickly. When they grows on the food, they convert the food particle, the organic material of food, into the poisonous material. And if we ingest that poisonous material into our body, that poisonous material may cause us a serious illness, and that serious illness is known as food poisoning. What happens, बच्चों, क्या होता है? Microorganisms, especially the bacteria and fungi, grows on the food, on the cooked food items which contain fats and oils, and that will result in the serious illness known as food poisoning. अगर बच्चे हम उस पॉइजनस फूड को खा लेंगे तो हमारे अंदर वो पॉइजनस सब्सटेंसेस, जहरीले टॉक्सिक सब्सटेंसेस आ जाएंगे, एंड वी विल सफर फ्रॉम अ डिजीज नोन एज फूड पॉइजनिंग. Characteristics of spoiled food, the characteristics of the waste food are the unpleasant smell. They gives a very unpleasant odor. एक बड़ी भद्दी smell आएगी, pungent smell, and they gives a very bad taste. Symptoms of food poisoning are vomiting. The food comes back out of our body, out of our stomach. Diarrhea. What is diarrhea? The excessive loss of water or the excessive loss of liquid waste out of the body through anus. That is known as diarrhea. Next, abdominal pain. There is a pain in this cavity. That is abdominal cavity. Below the chest cavity, there is a cavity known as abdominal cavity. Headache and fever. These are the certain general features of food poisoning. There are certain bacteria and fungus which majorly cause the food poisoning. The examples of certain bacteria, the bacteria which cause food poisoning are Salmonella, Staphylococci or Staphylococcus, and Clostridium. So the bacteria which causes food poisoning are Salmonella, Staphylococcus or Staphylococci, and Clostridium. And the fungal examples, the fungus which majorly cause the food poisoning are Aspergillus and Penicillium. As a large amount of food is spoiled by the microorganisms, that spoilage of food has various impacts on our lives, like economical impact, like food poisoning health effect, that is. So these impacts can be prevented by a method that is known as food preservation. माइक्रोऑर्गेनिज्म जो फूड को खराब कर रहे हैं बच्चे, उस खराब फूड से हमें बहुत सारा नुकसान है. क्या नुकसान है? पहला इकोनॉमिकल लॉस है, हमने जो फूड पर पैसे लगाए हैं वो खराब हो गए. दूसरा लॉस क्या है? हमारी हेल्थ का, क्यों? अगर हम वो स्पॉइल्ड फूड खा लेंगे, दैट विल कॉज अस फूड पॉइजनिंग. सो इस फूड पॉइजनिंग और इस नुकसान से बचने के लिए वी हैव अ मेथड, वी हैव सर्टेन मेथड्स, नॉट अ मेथड. That methods are known as food preservation methods. So first we'll discuss what is food preservation. The various methods which are used to prevent the spoilage of food are known as food preservation methods. The technique, the process by which spoilage of food is prevented by physical and chemical methods is known as food preservation. There are certain, certain conditions required to stop the growth of microorganisms on the food. Why we are stopping the growth? Why we are preventing the growth of microorganisms? Because these microorganisms are responsible for the spoiling of food. So if we stop the growth of those microorganisms, we can stop the food spoilage. We can stop the food poisoning. So now we'll discuss on what basis we stop the growth of microorganisms. The growth of microorganisms can be stopped first by the conditions become dry. In the dry conditions, the water content is low, and in the less water content, the microorganisms like bacteria and fungi do not grow. As we had studied, bacteria and fungi require moisture to grow. And if we make the dry condition, if we remove the moisture from any substance, from any food, then food preservation can be done. Second thing, the temperature should be low. The temperature is lower down. How? Refrigeration, bache. You know, we place our cooked vegetables and cooked food into the refrigerator. Why? Because refrigerator maintains the lower temperature. Refrigerator चीजों को ठंडा रखता है. And at that lower temperature, the microorganism growth cannot be able to occur. And due to absence of microorganism, the food spoilage is prevented. Next is the temperature being too high. If we boil the things, then also microorganisms get killed. अगर हम टेम्परेचर बहुत ज्यादा हाई कर देंगे तो हाई टेम्परेचर पर माइक्रोऑर्गेनिजम्स की ग्रोथ रुक जाती है. So the three conditions required to stop the growth of microorganisms is: condition should be dry, that is moisture free. Temperature is either too high or too low. Now we will discuss common methods of food preservation. We use various methods to preserve the food from its spoilage. The first method is chemical method. We use certain chemicals which preserve the food, and those chemicals are known as food preservatives. The chemicals which prevents the spoilage of food are known as food preservatives. The common food preservatives are sodium benzoate, citric acid, and potassium bisulfite. Sodium benzoate is used for preserving fruit juices and squashes. The squashes and the fruit juices which we used to drink in the summers is prevented by using sodium benzoate. They contain the sodium benzoate in it. Potassium bisulfite is used for preservation of cut fruits, the stored foods or the fruits which we are used to eat, the jams and jellies, etc. Second is preservation by salting. Salting is used from the ancient time periods. In the formation of pickle, the raw mangoes are first salted down and placed in the open area, placed in the sun, so that the salt will remove out the water from the raw mangoes and that water gets evaporated. And in the absence of water, the raw mangoes, which are turned to be pickle, doesn't get spoiled easily. So much is salting. What principle based on the salting is? Salting decreases the water content, decreases the moisture content, and due to that less moisture content, the food preservation is done. By salting, we can preserve the pickles. By salting, we can preserve the amla, which we used to eat, the amla candies. Those are preserved by salting. The next method, the third method, is preservation by sugar. Just like salting, just like the salt, sugar also lowers down the moisture content. Sugar also lowers down the water content. And the less water content, the growth of microorganisms is prohibited. The jams, jellies and squashes are stored for a longer period of time because they contain a large amount of sugar in it. And the microorganism won't grow in that large sugar content because of the less water content. The fourth method is preservation by oil and vinegar. The pickles are filled with oil or vinegar. Vinegar is sirka. Usually it is basically an acetic acid. The dilute acetic acid is known as vinegar. Sirka in Hindi. The oils and vinegar, the mustard oil especially, are used as a food preservative. In the case of pickles especially. So those items do not allow the microorganisms to grow and prevents the food spoilage. The next method of food preservation is pasteurization. Pasteurization is a technique which was discovered by Louis Pasteur in 1862. In this method, basically the milk is sterilized. But later on, this technique is useful for certain other liquids and certain other substances as well. What is pasteurization? Heating the milk for about 15 to 30 seconds at 70 degrees Celsius. At that high temperature, the microorganisms will kill. and then abruptly cool down, cooling it quickly at 10 degrees Celsius. That is used to prevent the reinfection. You know what, bache? In this technique, there are two separate points. First we heat it at high temperature. We heat it at 70 degrees Celsius. That is a very high temperature. That high temperature kills the microorganisms. Then we lower down the temperature, 10 degrees Celsius. From 70 we came 10. The low temperature, which is a very cold temperature. At this temperature, the reinfection of microorganisms is prevented. पहले तो हमने हाई टेम्परेचर करके माइक्रोऑर्गेनिज्म किल कर दिए, उसके बाद हम लो टेम्परेचर कर रहे हैं ताकि माइक्रोऑर्गेनिज्म की दोबारा से ग्रोथ, दोबारा से रीइंफेक्शन ना हो. So that method is known as pasteurization of milk. The milk which is pasteurized is known as pasteurized milk, and that milk is, usually sell out in the markets. The next is by freezing, by refrigeration. Refrigeration, cooling or freezing means we are lowering down the temperature. At lower temperatures, the growth of microorganisms is prohibited. If the temperature is too low, the microorganism won't be able to grow properly. And if microorganisms won't be able to grow properly, the food does not spoil. The food poisoning can be prevented. The food spoilage can be prevented. The next method is canning or vacuum packing. In canning or vacuum packing, first what we do is, first we sterilize the food. First we kill the microorganisms by heating it, by boiling it. Then that sterilized food is packed into the airtight packing or airtight containers or vacuum packing. Why is it so? Because vacuum means there is no air. And if there is no air, the spores of microorganisms, the reproductive units of microorganisms cannot be able to come in contact with the food. And that's how we prevent the growth of microorganisms. The next method is food preservation by heating. Usually in our houses we boil the milk, and that boiled milk will be placed in the refrigerator for the longer use. The boiling, the extreme heating will kill the microorganisms, and by that we can preserve our food items. The next and the last preservation method is preservation by dehydration. D means removal and hydra means water. The removal of water or the moisture content from the food items for its preservation is known as dehydration. Sun dehydration is a very common method. Certain vegetables like cauliflower, methi and spinach are dehydrated so that the water content is loosened away. The water content is removed away. Wheat and rice are also dehydrated for their longer preservation. So these dehydration methods are used, or these are the methods which we use for food preservation. Now we discuss the advantages of the food preservation. What is the benefit of the food preservation, bache? The first one is it decreases the wastage of food. As food is not spoiled, the food does not get waste easily. So the wastage of food can be prevented, and by the wastage of food prevention, we can get the economical benefits. We can get the health benefits. The next is increased the storage period of the food items. The food preservation methods increases the time limit for the storage of food. The stored food will remain healthy for a longer period of time, and that healthy food we can use later on. The next is nutritional value. The amount of nutrients in the food, the nutritional value of the food can be retained for a longer period of time, for a long period. The nutrients will remain intact in the food stored, in the preserved food. And due to those nutrients, we get the proper nutrient, we get the balanced diet. So, the third benefit is the nutritional value of the preserved food is retained for a longer period of time. The fourth benefit is the preservation of food ensures the availability of food to the distant places during any natural calamity or the places where the food crops usually don't grow, like the high mountainous regions. In those regions, the crops like wheat and rice won't be grown naturally. So, though food preserved can be approached, can be reached, can be taken to those parts, to those offshore parts, to those distant places where the food availability is very less. As well as food preservation also ensures the availability of food during the whole year. That means during the off season as well. As the preserved food will have the nutrient value intact with it, we can use that preserved food in the off seasons as well. So these are the following advantages of the food preservation. The first is it decreases the wastage of food. The second is it increases the storage time period for the food. The third, it ensures that nutritional value of the food is retained. And the fourth and the last one is it ensures the availability of food to the distant places and during the off season as well.